Okay. Hi, everyone. Thank you for coming today to our third or fourth. The number's escaping me. Fourth seminar. Thank you. So this is Aubriana. She is in the graduate school at Columbia, of course, and she's going to be talking about her research, which is understanding liver cancer. So before we start, though, I'm going to ask Aubriana just a few questions just for you guys to get to know her. So I guess we can start with how did you get started in science, Aubriana?
Yeah, well, when I was little, I loved collecting bugs and catching spiders, actually. My dad's house was full of bugs and caterpillars, and basically I spent a lot of time capturing them in like under glasses, and we had this really old like little tabletop microscope like from the 80s that my dad bought on eBay, and we would just like collect gross things and like look at them under the microscope basically. And I think like pond water and like looking at little like armor plated bugs were the craziest thing to me. Like I thought it was so cool um, that like there was so much going on that I couldn't see just with my eyes alone. And so I kind of fell in love with microbiology before I really knew what biology was. And so I really liked this pattern that I would develop of like looking deeper and deeper into what a cell is or what life is made up of, and like figuring out how these little pieces come together and make an organism or make an organ or a whole human. So that's kind of where what's like been constant in like leading me toward grad school here. And as a result, I think I do a lot of microscopy because I still love looking under the microscope at like the highest resolution I can and seeing all the tiny pieces that are in like a very small amount of tissue.
Nice.
Yeah.
Nice. So going off that, I guess in graduate school, what do you think is the best part of it? And then what do you think is also the hardest part of
Yeah.
your research?
The best part, like I've met so many cool people here who are just brilliant and awesome and wholesome and wonderful, uh, including the three board members here. That like even when I can't remember, I guess the hardest part is like making sure you're still motivated, like self motivated, and then the
Mm,
best part is like the people around you that remind you, like about the excitement that we all have. So even when in the harder moments where you might lose your motivation or get frustrated by something that's going wrong and like not seeing a result that you're expecting to see, um, you're able to come back and like chat with friends who are also like, yeah, I'm also struggling with this experiment that hasn't been working, but they're still excited in the way that you're excited, and it's being surrounded by that like constant like level of excitement in the background to understand bigger questions is like the best part, I think. Yeah.
nice. Okay, well, I guess I'll ask one more question and segue you into your talk. Did you always know that you were going to study cancer or
No,
liver cancer?
no,
Stumbled upon it?
not even not even as far as a year ago did I really decide that I was going to be studying cancer. I was especially interested in how cells die. So cells can die in, it turns out, a variety of different ways. And so I was especially interested in like how cells die before they're ready to in neurodegeneration, like Alzheimer's disease and or motor neurons in ALS, you know, where cells are dying before they're supposed to die. Liver is kind of on the opposite spectrum, where you have cells that should die but aren't dying. They're cells that are just growing like crazy, and you want to figure out how to tell them to die instead of how to tell them to be protected from it. So it's sort of two opposite ends of the same question of figuring out cell death.
Nice.
Yeah.
Cool. Thank you. So I guess we can let you get started. Everyone, feel free to unmute yourself at any point and ask your question, or if you don't want to do that, you can put it in the chat and we'll all be monitoring it so we can stop Aubriana and ask her. Yeah, okay. So take it away.
Yeah, awesome. Thank you so much, Natalie. So my talk is Understanding Liver Cancer. I'm a third year in the biological sciences department. And so I'm going to talk a little bit about what the liver is. So the liver is this big floppy organ that's under our lungs and above our stomach and intestines. It has a couple important jobs. So we have our intestines and our digestive organs right here next to it, sort of nestled underneath it. And so a lot of blood comes through, is absorbing nutrients from whatever we've eaten, absorbing water, and it has to undergo some filtration steps. And so you can think of the stomach and the spleen and intestines as digestive organs that are absorbing nutrients and filtering blood. And then our blood will actually flow up through the liver as though this liver is another big giant filter, and flow across the river back up to the heart, where it will continue to get pumped. And so liver is important for blood filtration, but also for sending materials back down into our digestive system. So there's a component called the gallbladder, sort of nestled inside here, that sends what's called bile. And bile helps us digest like dense fatty foods, basically, dense fatty materials that we've eaten that otherwise our intestines alone, just by themselves, can't really handle very well. And so it creates these salty, fatty dissolving chemicals that come from the liver and get sent down into our digestive system and help us absorb nutrients better. So it's very important. It helps take away toxins that are floating around in our blood. You might have heard that drinking a lot, like too much alcohol over a lifetime, can lead to damage of the liver, because the liver is doing a lot of the heavy lifting for filtering your blood of toxins. And so that can lead to sort of buildup of inflammation and fats in your liver. So it's a very, very important organ for maintaining our homeostasis and our overall balance. So again, blood sort of flows up toward the heart from our digestive organs down below the liver. They flow through these two avenues, the hepatic artery and portal vein. So hepatic really just is telling us that it's from or of the liver. So that HEP, HEP abbreviation, refers to the liver. And so you'll see the term hepatocytes too, that just means cells of the liver. So there's arteries and a vein bringing blood across this liver structure. And at the same time, in the opposite direction, we have bile moving out from the gallbladder down back into our digestive system to help us digest things. And so, as I said, I love zooming in as far as I can to figure out the substructure of things. So the liver is actually made up of tiny little repeating substructures called liver lobules. And they kind of look like a little bee's honeycomb in a beehive, because they're semi-hexagonal little blocks that fit together. And they just fill up the entire liver, well, not the entire liver, but almost the entire liver with these little repeating blocks. And so they're made up of a few common components, each little repeating block. It has a central vein, which has blood draining into it, and that central vein is leading back up to the heart, basically. So each of these little blocks has a little central vein that blood is flowing through. And then on the outside corners of it, it also has little tiny veins, and these are our portal veins. And so blood is flowing in from the portal veins and kind of soaks across down into the central vein, across these little spongy lobule structures that are made up of hepatocytes. So what you're seeing on the right side here is here where I'm tracing with my mouse is like sort of the boundaries of one of these little honeycomb hexagons. And you can see a portal vein here, and the blood would actually sort of flow. It's flowing up toward us, like through the screen. And it's also flowing across these little cells here in the middle and draining down into the central vein. And so the blood is actually getting filtered across each and every one of these little lobule structures, which is really, really cool. Oops.
Is it
Oh, sorry.
A process that is energy free? Is it like a process of diffusion, or is it kind of just due to gravity, it just kind of falls into the central vein?
It is partially diffusion, but there's also the partial pressure coming from, so there's blood pressure within the vascular system. So that's a great question. It's actually also powered by this positive pressure from these portal veins moving toward the central vein. So you can imagine if the pressure is lower at the central vein, the natural drainage would go toward the central vein.
I see. Thank you.
Yeah. This is to emphasize that we have these little repeating structures and filling up the liver and helping it do its function. And what fills in this middle part is a couple different types of cells. So we have hepatocytes, which make up about 80% of the cells in your liver. And they're these sort of big, blocky looking tan cells here. And we also have sinusoidal cells, which make up the actual little winding veins here, you can imagine. So you have cells sort of tracing between the hepatocytes that allow for this flow of nutrients. I won't go too into detail of this, but this is to emphasize, this is where our liver sits on top of our stomach, our pancreas, and then our intestines are down below here. And here's our gallbladder draining bile down into our intestines, actually, directly into our intestines, where our food went. And this is a sort of a 3D image of the little lobule structure that we just saw. But where you can see that central vein, which just looked like a little circle before, but it's actually flowing upwards toward the vein, toward the hepatic vein, and toward the rest of the heart. And these are the sinusoids sort of tracing their way through the lobule structure between all these big hepatocytes. And so why do we care? Why do we even care what this looks like? Well, things can go wrong. And the thing that I study that goes wrong is liver cancer. And so there are a couple different things that can lend to a higher risk of liver cancer. And the main ones are drinking too much alcohol, smoking a lot. So these two cause your liver to do a lot of work that it wouldn't necessarily need to do to filter your blood and filter toxins out of your digestive system, and try to protect the rest of your organs from contaminated blood. And also too much fatty content inside your liver can lead to something called cirrhosis, where you have a lot of bubbling of lipid cells and fatty cells that increase your risk of getting cancer down the line. And there are also particular, something that's special about the liver is there are also liver-specific viruses that humans can get. And the big two culprits of these are hepatitis B and hepatitis C. So you might have heard of these if you've gotten shots at the doctor, or sometimes if you're working at a university, they will offer immunizations for hepatitis B, for example. So these are in hopes of preventing infection with these, because they can lead to a severe infection in your liver that leads to a higher risk of getting liver cancer down the line. And so what do you do once you have cancer in your liver? Well, that's a big problem, but that's a problem that a lot of people have to combat with in the moment. And so we want to understand at a very small level what's really going on here. So if you're looking at a liver, it has a tumor growing inside of it. What a tumor is a little bundle of cancer cells. Cancer cells are cells that are just dividing and growing when they shouldn't be growing. They have passed their regulatory checkpoints. They're ignoring the signals that say, wait, you can stop growing. You've filled up the space that you need to. They're ignoring all of those instructions. They're going ahead and dividing anyways until they build this unregulated mass. It doesn't have a nice structure the way our organs do, because it's not communicating with the cells around it properly. And so it builds and builds into this ball of cells. One dangerous thing that can happen and that you might hear about when people talk about cancer is metastasis. And so what you start off with is a tumor in the liver. That's a primary tumor because it started in the liver and it stayed in the liver. One dangerous thing that can happen is that a cell breaks loose. A cell can break loose from this initial primary tumor and start moving around. And it can make its way into the bloodstream. And if it infiltrates its way into the bloodstream, it can do one of two things. If it's a metastatic cell, that means it's able to break past the vasculature and colonize a whole new organ. And so even though you maybe only started off with a tumor in the liver, if it progresses and cells escape and move through your blood into and find a new place to make their home, like in the lungs, you can have a metastatic tumor, which is the result of this metastasis moving from the liver through your blood into a whole new organ system. And so that can lead to having tumors in different places of your body, which is very, very dangerous. They don't necessarily have to do this. So they can be a non-metastatic cancer cell that breaks through into the bloodstream, but doesn't actually colonize or find a new place to settle. But doctors are always really worried about metastasis because it is possible and it can be very, very difficult to treat a cancer in multiple organs as opposed to a cancer just isolated in one organ. Okay, and so this is a little, I want to give a content warning just for the next slide and I'll announce when it's over, but I wanted to show what a tumor actually looks like when it's in a liver. Okay, 3, 2, 1. This is part of a mouse liver, and so these white nodules here are actually these blobs of cells that have grown without regulation and grown into a tumor. And so they're sort of placed randomly in this organ because they started semi-randomly and they grew without the rest of the organ putting enough stressors on it to sort of kill it off early on. Okay, the gross picture is gone. You can open your eyes again. And so this is a huge problem, particularly liver cancer is a huge problem because even though most cancers are going down in incidence as we figure out more and more about them, liver cancer is actually the one that's increasing. So we like to think in the medical field like everything is improving and our treatments are getting better and better and diseases are getting solved, but actually liver cancer is especially worrisome because it's getting worse, globally at least. And this is a global map of the mortality rate from liver cancer updated in 2019. It's especially bad in parts of Northern and West Africa, Eastern Central China, and the Eastern Asia, or Southern Eastern Asia, sorry, and the Northern American continent. And so it's something that we should be very concerned about and something that I definitely personally want to understand better.
Brianna, we've got a question here.
Oh.
When you say it's getting worse, do you mean in terms of a rise in cases, or it's getting worse because it's getting harder to treat?
A rise in cases, yes. So it's increasing in a rise in cases. So it could be because of the spread of risk factors like HBV and hepatitis B virus and hepatitis C virus, and also increase of humans overall consuming more alcohol perhaps on a regular basis, and potentially smoking more than centuries ago, past generations. Yeah. But it's unclear exactly, like it's hard for us to pinpoint exactly just one reason why we're seeing this increase. And if the treatment could keep up with this increase of incidence, we wouldn't quite have this increase in mortality reflected, right? So what I'm going to talk about next is how we're limited in ways to treat this as well. It's also a disease that's hard to detect early on. So for example, some cancers are easier to detect early on because you get early symptoms or signals that something's wrong, that something's going on, or you might have sort of early feelings of sickness related to the development of a tumor. But it's oftentimes harder to detect liver tumors until they're a significant size already. And not many people do satellite programs or screenings generally without worrying about something already to see if there are any tumors developing in their livers. Yeah, any other questions?
Thank you.
Awesome. So as I said, I love to zoom in. So liver cancers don't always look the same necessarily. They can start in different places in these substructures, right? So they can start in the hepatocytes, which is the most common one. So if a cell here gets screwed up and undergoes a mutation and starts proliferating like crazy, that could lead to what's called a hepatocellular carcinoma or hepatoblastoma, where you're getting a mass derived that originally started in a hepatocyte. But that's not the only place you can start a liver cancer. You can also have it originate in a vessel structure. So it's also possible for you to have a liver vessel structure cell become mutated and have that be the origin of a tumor. And you can also have it originate in bile ducts and the cells of bile canals. And so that's what's called a cholangiocarcinoma. And I'm particularly interested in this first one where it's originating in hepatocytes and understanding how a hepatocyte that's growing and in a tumor environment is interacting with the cells around it, and how the cells around it might either better attack it or better protect it from treatment.
Adriana, we have a question here. Do we know which structures are I'm on mute. Do you know which structures are more likely to become cancerous?
Yeah, so the hepatocytes are more likely to become cancer. Part of it is because there's so many of them. So 80% of the cells across your liver are going to be hepatocytes. This first one here. They just make up a lot of the liver, and so
so like statistically, almost they're more likely, right?
Yeah. So if something gets messed up, it's most likely going to be a hepatocyte first. But there are definitely human cases of cholangiocarcinoma and angiocarcinoma. They're just more rare. Yeah, great question. Okay, so let's say we've developed a form of liver cancer that started in the hepatocytes. What actually happens to patients at different stages of the disease progression? So currently, what happens if you have early detection? So say you had a scan of your liver done in the hospital, and they say, oh, we found a small lump buried inside of part of your liver. We're worried about it because based off of the markers associated with it, or perhaps a biopsy or markers in your blood, we think that this is a serious, or what could become a serious tumor, a hepatocellular carcinoma tumor. And so what they'll do is actually do what's called resection or surgery, where they literally open you up and cut it out as best they can without doing extra damage. If it's really, if it's severe, they can also do what's called a liver transplantation. And so that's where patients can enroll for liver transplants in the case where someone else can donate part of their liver, or there's a donation, sadly, from another accident or something like that. But this is early stage, and these are also pretty intense operations for just early stage, right? So what about intermediate? So there, if it's intermediate and it can't undergo resection surgery, often the next best option is drugs. And so there are a couple of drugs on the market, particularly sorafenib. Unfortunately, patients don't have a great response rate to sorafenib. And oftentimes they also get bad side effects. They can feel really, really sick, or they can have, they have bad side effects that lead to doctors usually taking them off the drug, perhaps before they see significant shrinkage of their tumors. If it's further advanced, they might do a combination with not just drugs and chemotherapy, but targeted chemotherapy. So chemotherapy is basically where you send in cell-killing poisons into the body in hopes of killing your tumor cells. And so often it makes the patient very sick as well. So one approach that doctors are trying to take more often now is targeted chemotherapy, where they literally send a little teeny tiny catheter tube into your veins and into your vasculature, or sorry, actually into your arteries, to try to send in chemotherapy holding beads and sort of fill up the arteries and capillaries that are feeding this tumor, and try to sort of sequester it and have most of your chemotherapy end up here and around where the tumor is being fed by fresh blood. And so that is a very promising methodology as well. But apart from these options so far, once it's at a terminal stage, patients are left with best of care approaches. And unfortunately, there aren't many other options except for making their lives as comfortable as possible as the disease progresses. So
Brianna, I've got, sorry. Can I ask a quick question we have in the chat?
Yeah, totally.
So why is the liver capable of functioning with only some sections remaining?
Yeah, the liver is amazing. It is very, very highly vascularized. So it's not just sort of, as long as you're not disrupting the initial input and output from your, like to your hepatic vein and from your portal veins and hepatic artery, it can function with a piece taken away from it. And it also has some amazing healing regenerative abilities compared to other organs like the heart. But like taking out chunks of it is not super detrimental to the patient. Sorry, do you see the question again? I think I got off track maybe.
No, I think you're right on track. Just like how is it able to survive with pieces taken out of it? So I guess it's like the vascularization, right?
Yeah, it's highly vascularized. So even if you're missing like a part of it, the rest of the liver can still do its job mainly, right? So it won't have as terrible as effect as opposed to like if you took out a piece of the heart, right?
Right. Yeah.
It's much more disruptive. So and also the beauty of the liver is it's, I don't want to say squishy, but it's kind of like squishy and amorphous in that like the repeating lobules are throughout the entire liver. And so a lot of the liver is doing the same, like similar work across the entire structure. Yeah.
You mentioned there was some sort of kind of ability for the liver to heal itself. Is that because the cells have some sort of capability to differentiate even once they have a role?
Yeah, so there are actually hepatocyte progenitor cells. So every organ sort of has a base level of progenitor cells. They're ready to replenish cells that get turned over or get lost in injury. Sort of like how our skin cells constantly replenish themselves. That's because at the very, very lower levels on the inside of our skin, we have these progenitor cells and basal stem cells that constantly differentiate into more recognizably skin cells. And so that's the case for liver as well. You also have progenitor cells, and you also have vascular progenitor cells. So for example, if part of the liver needs more nutrients or needs more vasculature, it can send out signals and communicate with other parts of, or cells can communicate to one another through signaling and indicate for vasculature to grow toward it. So that's, I won't go too far into that, but yes, so cells can communicate when they need wound healing, basically. So macrophages are also pretty instrumental to that in healing and then helping bolster regeneration of new progenitor cells into more differentiated hepatocyte cells.
Thank you.
Yeah.
Brianna, I have one more question from the chat here, and then we'll let you go forward. So right here, we have someone asking, why is alpha fetoprotein elevated in hepatocellular carcinoma?
Oh, dang, great question.
I know, that's above my knowledge level.
Actually, yeah, there are some, oh wow, everyone's doing my PhD for me. There are some subtypes of cancer cells, or sorry, liver cancer types that have elevated alpha fetoprotein. So I talked about how you can have cancer from like the bile ducts versus the vasculature versus hepatocytes. So it's the ones that are derived from hepatocytes where we see elevated alpha fetoprotein levels. Let's see. The mechanisms, as far as I know, aren't super clear from like case to case. But I can add some links in the chat if that's all right. Does that sound okay? Like sourcing to papers that explain it better than I can.
It seems like a complicated question, so makes sense.
Yeah.
Thanks.
Awesome. Yeah, really good question. And it is the foremost marker that people use currently in the field. So to get like an early diagnosis of HCC progressing. Yes, so what I'm especially interested in is resistance and understanding. So even if we hit tumors with drugs, we tend to have high incidences of resistance, unfortunately. And so there are a couple ways that, in general, tumors can resist treatment. One way is that they can already have resistance built into them, which is called intrinsic resistance. So say a couple of them are just already have the mechanisms inside of them. They were never going to get killed by this drug. Their protein target where the drug would have bind and disrupted things is modified in such a way that it would never bind that drug at all. Even though maybe 90% of the cells will die, if 10% of the cells survive in that initial tumor, they can survive and then regrow into a new tumor as well. So, and that's an unfortunate form of tumor resistance. Also, even if in this case, every cell is targetable by a drug, say, it's possible that they can adapt and change their behavior or change how they express proteins inside of them in order to sort of get around this cell death inducing drug. And so they can change and modify their expression and become resistant in that way, and then recover the same level of growth that they had before. A third mechanism, or third way to have resistance is what I'm most excited about, which is where cells talk to each other. And cells make contacts with each other and listen to one another and send each other little messages in the form of protein signals that help them as a whole tumor together resist a drug. And that's what I'm especially excited about in terms of understanding how these intermediate and advanced stage tumors can resist drug treatment. And so if we zoom into a tumor, I know earlier I showed a tumor as like all the same cells, but that's actually not necessarily the case. You can have tumors that are built up of a bunch of different cells that sort of grew together and are mashed together And a tumor can have infiltration of a bunch of different type of cells, and you can have these cells communicating with one another. One thing, for example, that's been found is that tumors that are surrounded by these pink cells, these stretchy fibroblast cells, actually do better because these fibroblast cells can protect these inner cancer cells by feeding them growth factors that help them grow better and just overcome the outside stresses of growing in this constraining, limiting liver tissue. And so I want to better understand what's going on in here. What are these cells really saying to one another? And like why, if you have certain compositions or certain combinations of cells, can they better resist our drugs? If you're missing one or two of them, does that mean they're sensitive to drugs again? What does it take to sort of break down this structure and this, this sort of community of cells protecting one another? Any questions about that? Nope. Cool. All right, I'll keep going. And another thing that can happen to cells is they reach out and draw nutrients toward them, and they reach out through these communications and tell other cells of the vasculature to grow toward them and give them fresh blood. They're like vampires. They're completely like vampires. And so they cause veins and arteries, or sorry, they cause arteries to grow toward them and bring new nutrients toward them. And so that also helps make them resistant to drugs. So tumors that are more like this, that have a better nutrient system and better vasculature, can often survive treatments better, unfortunately. And another thing that they can do is they can sort of, I talked about these communications together, but cells can sort of hold hands with one another through these junctions between them. And one that I'm especially interested in is called ecadherin. And you can imagine, I don't know if anyone's played the game Red Rover when they were little, but you sort of have two lines of kids holding hands with each other. And you say, Red Rover, send Angela over, and then Angela tries to run as fast as she can and sort of break through the hands. So you can imagine if nobody's holding hands with one another, it's much easier for Angela to break through the line. This is sort of what we're seeing that cancer cells can do as well. They can have these tight connections to one another where they hold tightly to each other and resist outside stressors to better resist treatment. I'm realizing, and this might be a little bit much, but this is kind of just demonstrating exactly that. ECAD is one of those junction hand-holding molecules where cells can touch each other and make this connection to one another. And we see that for the cases where they're not really touching each other very much, where they don't have a whole lot of hand-holding to one another, they undergo death much more easily when we hit them with a drug. And so on these cells were all hit with a drug down here, where you see this green lighting up, and the green is an indicator that these cells are currently dying. And so you can see, even though these were all hit with the same amount of drug, the ones that are very tightly packed and touching a whole lot of other cells and have a whole lot more of these junctions together, resist the drug. They survive under the same treatment conditions, whereas the ones that are alone or in very, very small packs, they succumb more readily to the treatment. And this is saying the same thing. Basically, these are cells that are sensitive to a drug, and these are cells that are resistant to a drug. And we see that the ones that are resistant are especially high in the level of ecadherin, or this protein that binds these cells together better. And so I'm interested in doing this. This was not done in liver cells. This was done in gut epithelial cells. But I'm really interested in doing this for liver cells as well. Yeah. And so this is kind of what it looks like when I do early tests of our sorafenib. drug that's used in the clinic on liver cell lines. This is very early data, but what you're seeing here is a couple versions of liver cancer cell lines that were taken from human beings. And then after some treatment with the drug that's currently used in the clinic, and we do see that they start to die. Oops. And it seems as though, just looking at this early on, the ones that are tightly packed together seem not to succumb as early as the ones that are isolated and on their own. Yeah, and so what I overall am interested in is understanding what it takes for cells to be resistant or especially sensitive to drugs. Is it the way in which veins can come and bring new nutrients to them and feed them with fresh blood? Is it about how much ECAD and like how many of that hand-holding interactions they have by ECAD-herin junctions? Are any of these factors what allow liver cancer cells to persist through treatments? And so I'm interested in looking at this as a readout with mRNA. So in our cells, our genetic information starts as DNA. Every cell has a blueprint called our DNA, which encodes every protein that we could possibly need. And pieces of that DNA get turned into mRNA, which later get transformed into proteins. And proteins are actually what we read out as like the enzymes and proteins that build the necessary machinery in our cells and distinguish cells between one another, between different types. And so targeting mRNA is what I do in the lab, and this is basically what I'm looking at in the lab on a day-to-day basis. So this is a picture of healthy liver, not cancerous liver yet. But what I'm able to do is tag individual pieces of this mRNA and learn the different identities of individual cells at the single cell level inside of these liver lobule structures. And basically what I want to be able to do is compare images like this to images taken from cancerous tissue and see how different cells interact with one another and where they're getting disrupted and where they're losing their ability to communicate and send signals to one another. Yeah, I think that's all from me. Oh, that was a long time, sorry.
That's awesome, Aubriana. That's really cool. Pretty pictures. Does anyone have any questions they want to ask or put in the chat? If not, feel free to log off.
Yeah, thanks everyone for being here.
Yay. Aubriana, I have a question. That last image, what type of microscopy is that?
Oh yeah, that's
is it confocal?
Yeah, that's
is it more
disc confocal.
Gotcha.
Show it again. Let's see. Yeah, this is a 60x image targeting 3 different genes in 3 different colors. But yeah, it's a spinning disc confocal. Basically, you can do like scanning confocal, I believe, where it like goes pixel by pixel by pixel and sort of goes down the line. This one is really nice because it goes really quickly, where it's sort of like the whole field of view at once and sort of builds it up through the Z stacks, which I really appreciate. So it's much faster.
Cool.
For the microenvironment of the tumor, you have kind of a key on the side for all the tumor cells. Do we know the functions of each of them already? Is all we need to discover kind of the interactions between them, or do we not?
Here? Is this what you're talking about? Yeah. So CAFs especially have been pretty well characterized in liver cancer. This image is not specific to liver cancer, actually. So this is a sort of a general image of tumor microenvironments. You also see similar complexity inside tumors for like breast cancer tumors, for pancreatic tumors. So this phenomenon is not just happening in liver cancer, but and the interactions between these different cells are better characterized in some cancers than others. So far for liver cancer, I believe they've seen particular significance for T cells and regulating T cells, definitely tumor-associated macrophages. So those are macrophages that are found in the tumor microenvironment as well. And let's see. These are referring to just like necrosis and dying hypoxic cells that don't have enough oxygen. Yeah, I think natural killer cells have been shown to have significant interactions as well. Yeah.
Thank you.
Mm-hmm. And also the vasculature as well, I'm a little more familiar with. So there's an interaction that goes on here. So there's a particular factor called vascular endothelial growth factor that gets secreted out from especially resistant tumors, actually from tumors in general, in hepatocellular carcinoma, but it can signal out to other surrounding vasculature or like the cells in the nearby arteries, that they should grow out toward the new tumor and build new branches to sort of reach out and touch the new tumor and help it get more nutrients. And so that's a signaling factor that's super important for new tumors. Otherwise, they might never get the nutrients to grow beyond a very, very small size. Mm-hmm.
Do we know what sort of signaling kind of techniques tumors use to cause this, or not really?
Yeah, they can secrete it out from, so a cell can like package secreted proteins and actually like push it out into the outside lumen. I believe you can see somewhat like gradients across cells as well of different secretion factors, but they can literally secrete stuff out that diffuses and lands on receptors on other cells. And so an endothelial cell way out here that makes up part of an artery might have a receptor ready to receive the growth factor from the tumor, but it's not until a tumor develops and then sends out that signal that has to land on a receptor, get caught, and then tell the rest of the cell to grow and tell the surrounding cells to also grow, that you would actually get this phenomenon of the building of new vessels.
I see.
Mm-hmm.
Have techniques been tried to be, what am I trying to say? Have techniques been kind of developed to ensure kind of the kind of protein signals from the tumors not actually bond with the receptor cells from the veins to ensure that they don't get the nutrients and so that therefore the tumors die quite early on?
Actually, the drug here, sorafenib, actually also partially inhibits that receptor that I was just talking about. It somewhat prevents the signaling after that receptor receives the signal. It's a kinase inhibitor, and so that receptor actually has a kinase domain that then has to communicate to the rest of the cell. Okay, we got to activate, we got to grow, make new vessels, right? So one thing that the drug is intended to do is stop that kinase module from telling the downstream sort of chain reaction to continue, to sort of stop it there and not lead to this growth of new vessels.
You see, thank you.
Yeah.
Cool. Does anyone else have additional questions? Okay. Well, I guess, yeah, everyone, thank you for coming then. Adriana, thank you. That was really interesting. Yeah, we'll have another one of these in a few weeks, everyone. So just keep an eye on your email. Good to see you all.
Awesome. Thanks so much. Thanks.
Bye, guys.
Watching.